Okay, today we're going to talk about plant biotechnology and tell you part of a long ongoing story of one of the most famous GMO crops, which is golden rice. Okay, first, most people don't really realize that genetically modified foods are extremely prevalent in the United States and in some other countries as well. That the vast majority of crops, like corn, soybean, etc., are in fact genetically modified in ways to encourage them to resist pests and especially to be resistant to pesticides, as we'll talk about next time. Okay. However widespread this is, the skepticism of the world really about the safety of GMO foods is incredibly widespread. Right? This was a study done by the Pew Research Center in 2020, where they asked people whether they felt, different countries, whether they felt that genetically modified foods were unsafe, safe, or that people didn't know. And there was no country in the world where people agreed universally that genetically modified foods were in fact safe. The more honest people said they really didn't know. So even in the United States, fully a third of the population feels that the genetically modified foods are unsafe. But they're eating a lot of corn, so go figure. Okay, so what I want to do today really is to go through how genetically modified foods are made in general and to give you some feeling for what's actually in there that makes them genetically modified, transgenic, or non-natural, depending on your point of view. Okay, so we're going to talk mostly today about Agrobacterium-mediated transformation of plant cells. Agrobacterium has been used to modify most of the crops that we will be talking about. But the other method is biolistic transformation, in which the DNA to be transferred is actually literally shot on little metal particles through a gun, deposited in the cells, and the cells that survive will somehow magically acquire the transgene. Okay, we'll talk more about the process after we've done the basic manipulation to introduce the DNA into the plant. We have to then grow the plants under selection and then actually regenerate an intact plant and breed it before it can count as truly genetically modified. So we'll talk about Agrobacterium in some detail first. Okay, so Agrobacterium Tumifaciens is a bacterium, lives in soil, and it parasitizes plant cells. Like the natural organism actually creates tumors in plants to create an environment for itself to thrive. Okay, so this capability of Agrobacterium is encoded on a plasmid called the TI plasmid. And what the Agrobacterium does naturally to the plant is transfer a section of the TI plasmid to the plant nucleus, where it becomes activated and synthesizes proteins that the bacteria will find helpful. And, well, it produces actually strange molecules that the Agrobacterium finds helpful, and also creates a condition where the plant cells grow essentially an undifferentiated mass in essentially a tumor on the plant. Okay, so here's an example of a crown gall tumor caused by Agrobacterium tumifaciens. So it consists of a large mass of largely undifferentiated cells in which the Agrobacterium stick and thrive and are provided with essential nutrients by the plant. So the Agrobacterium causes the growth of these tumors as well as causing the bacterial cell to provide essential nutrients to the bacterium. Okay, so here's just more than we need to know detail of what happens when interactions occur between the TI plasmid and the plant. And you don't need to know all the bits and pieces of it. The important thing is the TI plasmid exists in the Agrobacterium and that it contains genes that are on the plasmid itself, but also a section of the DNA which gets transferred to the plant. Okay, the part that gets transferred is called the tDNA, and what it does, it goes to the plant nucleus and then expresses the genes that the bacterium are going to find helpful. And so, of course, we're going to synthesize auxins and cytokinin in order to produce the tumor, to produce a mass of undifferentiated cells. But we're also going to produce an interesting class of compounds called openes which the bacterium will then take up and be used for nutrition for the bacterium. Okay, so here's a very schematic view of the TI plasmid, and it emphasizes some important features of it. The region that's going to be transferred to the plant extends from what's called the left border around to the right border. So those are the ends of the tDNA, the part that gets transferred as a single strand actually. And the whole molecule, of course, contains other things as well. So the part that gets transferred to the plant includes the auxin and cytokinin genes, as well as the opine biosynthetic genes to cause the plant to produce these novel compounds for the benefit of the Agrobacterium. Then we have, of course, in the TI plasmid itself, genes that enable the bacterium to use those molecules for its own nutrition. Of course, we need to have an origin of replication so that the plasmid can replicate in the Agrobacterium. But importantly, there's also a vir region or virulence, and that encodes the proteins that are necessary for transfer of the tDNA to the host, to the plant. So we don't need to transfer the virulence genes, but what you do need to have is the virulence genes in the plasmid so that the tDNA can get transferred to where it wants to go in the plant. Here's a slightly less schematic version of the whole thing, but it does emphasize the important features of the molecule. The genes required for tumor formation, for sure, the left and right border, and the opine catabolism, and the vir genes. Okay, so that's a slightly more detailed view of what's going on with this huge plasmid. Okay, so the left and right borders are important in that, for our purposes, anything between the left and right border that we put there is going to get transferred to the plant. Okay, so this is not the natural part of Agrobacterium-mediated gene transfer. This is not what Agrobacterium wants to do, but that's what we're going to want to do. But as far as the Agrobacterium is concerned, that's the part that's going to promote the plant cell proliferation and also to cause the plants to make the opines. Okay, so these are unusual molecules. They're related to amino acids, and that the Agrobacterium can use them for nutrition, nitrogen and carbon. So the plant cell is going to feed the Agrobacterium under these circumstances. So here's what opines look like. They look somewhat like amino acids, and they are synthesized by the plant cell and then secreted in such a way that the Agrobacterium can then take them up and use them for its own nutrition. Okay, so the part that is not transferred includes vir. Okay, and that's going to include binding to the tDNA, endonuclease, delivery to the plant genome, etc. Okay, so how we're going to make the TI plasmid work for us, we're going to use the tDNA to transfer our gene of interest into the plant cell. So anything between the left and right borders will be transferred, and we're going to need to keep the left and right borders for our purpose. But in terms of using these as cloning vectors, etc., or for plant biotechnology, we need to get rid of the tumor-promoting parts of the plasmid, and then we're going to keep the useful parts. So we're going to get rid of the auxin, cytokinin, opine biosynthesis genes for sure, but we're going to also have to add in some selectable markers and whatnot to make this a good cloning vector for the usual purposes. Okay, so here's a maybe too schematic version of a plasmid that one might use for this purpose. We have the left and right borders, but instead we have any gene of interest cloned into what was the tDNA that contained the auxin genes, etc. We also need to include an antibiotic or herbicide resistance gene to select transformed plants later on. We need to have either in this plasmid or in another plasmid in the same cell the virulence genes that encode the nucleases, etc., to get the tDNA to go to the plant. We don't need the opine catabolism genes anymore because we're not going to ask the plant to make those for us. So that certainly doesn't need to be there. A slightly different version of this is a little more realistic. We have a multiple cloning site, which isn't really shown here, next to a promoter, where we're going to then insert our gene of interest, our selectable marker that's going to be used for the plant, origin of replication for Agrobacterium for sure, but also an origin of replication for E. coli, because we're going to want to do our cloning in E. coli for many real reasons, but partly because the TI plasmid itself is too big. We also need selectable markers for transformation of E. coli and also the Agrobacterium, but kanamycin can be used for both. Okay, so notice this particular plasmid does not contain the Vir genes, which, as I said before, are actually needed to get the tDNA to go to the plant. Well, all right, so we need a promoter, etc. There's also a multiple cloning site. All right, so the gene of interest is going to be put in there, and the full-size tDNA does have the Vir genes on it, but it makes the molecule a little bit cumbersome to work with. So many times the Vir genes are supplied on a second plasmid in the Agrobacterium, and that's considered a helper plasmid. So the Agrobacterium that's going to transfer the tDNA to the plant needs to have the Vir genes in it, but it does not need to be on the same molecule as the part that we're going to transfer. And we certainly don't need the Vir genes when we're doing our cloning in E. coli. Okay, so we're going to transfer the tDNA, having made our clone DNA with our gene of interest, with our promoter, with our selectable marker, etc. And it's in our Agrobacterium with a helper plasmid if needed. And we want to then introduce that tDNA into the plant. So this is strangely simple, actually. You co-cultivate, or basically let the Agrobacterium grow with pieces of plant. And the agrobacteria will then infect the wounded plant cells, which can later then be cultivated more and regrown into transgenic plants. Okay, so what you can use as far as plant tissue, some people just use leaf discs, like just get a paper punch and chop out a piece of disc, and you're going to have ragged edges that the Agrobacterium can use to infect. You can use cuttings of roots, you can use plant embryos, and you can use dedifferentiated culture cells that are formed what's called a callus. So there are many options for how you then incubate your Agrobacterium so that the tDNA can do its job. Okay, so in terms of selection, we need to have our gene of interest, but we also need an antibiotic resistance gene that we can use to kill the plant cells that did not get the plasma, did not get the tDNA. Because not all the plants will actually get the tDNA. You want to have a way to allow those cells that did get it to grow out. And those cells will grow out around the edges of the leaf disc or around the edges of the callus culture, showing that they have actually acquired the antibiotic resistance and therefore very likely the gene of interest. So then they're placed on growth medium and allowed to grow out. Okay, so plants are really relatively easy to regenerate. You can generate whole plants naturally, whole plants from many, many different types of plants and different types of tissue. Carrots are the most famous for this, being the first and oldest and easiest, but this can be done with many, many, many different kinds of plants. Okay, so for example, rice, when you're growing cells in a growth medium, that they grow into these sort of masses of undifferentiated cells, and that by switching the medium to various growth-promoting, shoot-promoting, root-promoting hormones, a complete plant can be regenerated from these non-differentiated cells. So here's different pictures of callus, rooty callus, shooty callus, etc. Okay, so eventually you have your callus cultures, and they are grown in appropriate medium to encourage them to root, to form shoots, and then they're transferred, and eventually they will fully regenerate a complete plant, which should be transgenic as a result of our Agrobacterium manipulation. So once you get the new plants, these are considered T0. They've been selected to carry the tDNA, but the tDNA is integrated at different locations in the plant genome. The plants are likely heterozygous for the tDNA, and because the location differs, the plants will express different levels of the transgene. So the entire process is going to involve growing up the plants, regenerating the plants, getting them to be happy, and then crossing them, largely self-cross, in order to get homozygous plants that actually contain the transgene. Okay, a long, sad story, or a long, ongoing, and very fascinating story, the example GMO being golden rice. Okay, so the picture shows a picture of regular rice and the first generation of golden rice, which you notice is a little bit yellow. The current generation of golden rice is very, very yellow. Okay, the reason it's golden is it produces a lot of beta-carotene. This is a compound that is converted to vitamin A in our bodies, and vitamin A deficiency is a leading cause of blindness and mortality in low-income countries. So the idea of this golden rice project, which started back in the 90s, was to create rice which itself produced beta-carotene, which would be converted to vitamin A, in order to prevent the vitamin A deficiency that is so important in poor countries. So vitamin A deficiency, retinol is the same as vitamin A, and beta-carotene is the product that we're going to try to make in the plants because it's going to be converted to vitamin A in our bodies. Okay, so here's what vitamin A looks like. It's the same as retinol, and it's made from beta-carotene, which is orange. Okay, so vitamin A deficiency. Vitamin A deficiency is rare in high-income countries because we do have a diverse diet, although it does occur among kids that won't eat vegetables. So good sources of vitamin A include green leafy vegetables, tomato, orange and yellow vegetables, fruits, etc. So because of our diverse diets, vitamin A deficiency is relatively rare. But for people in poor countries who do not consume these items frequently, where their main diet is rice, they don't get enough vitamin A in their diet to avoid a deficiency. And that's actually tragic because of the consequences. So I got this slide from NIH as far as what vitamin A is supposed to do. The connection to blindness is that vitamin A is part of the rhodopsin light-sensing protein in the retina. And in the absence of vitamin A, the vision diminishes considerably. But it's also required for differentiation and function of the other membranes in the eye and the cornea. So extreme vitamin A deficiency will lead to blindness. It also has many other roles in the body involving growth and development, such that if you're deficient, you're much more susceptible to mortality due to other diseases, including things like measles. So the deficiency is a severe health hazard. Okay, in terms of prevalency, it's almost 50% among children up to 5 years in sub-Saharan Africa, South Asia, etc. And so the severity of the diseases and diarrhea and mortality that results is truly a problem. It is a leading cause of blindness worldwide. Okay, so the idea was to engineer the rice to make beta-carotene so that rice in the diet would be a sufficient source of vitamin A to avoid the basic mortalities and illnesses that result from this deficiency. And this process has been going on for a while. Okay, so the first version of golden rice actually came out in the year 2000, and the rice that were made were successful in producing beta-carotene, as envisioned by the very pale yellow color of the rice. But the levels were too low to be useful, so later derivatives, especially golden rice 2, came out in 2005. This rice was successful in that the levels were 30-fold higher than in the first version, and later, because of this, it has been approved for use in the United States and Canada, and just recently in 2021 in the Philippines, but in no other country has it yet been approved for use. All right, so what was done was engineering the beta-carotene pathway. Rice itself has the enzymes to make beta-carotene, but they're not expressed in the endosperm. The endosperm, of course, is the seed, the part we eat. And at the point people started this work, it wasn't even known which precursors were present in the endosperm, as to say, what enzymes you would even have to start looking for to put in to make your beta-carotene. So it was a huge amount of work to get this sorted out, to figure out what was going on, and to identify genes that could be used for the purpose of making the beta-carotene in the rice. Okay, so the first version of golden rice included the source of the gene, the first one, the phytophytoene synthase from daffodil, then a bacterial enzyme which encodes phytophytoene desaturase, which actually is responsible for two steps in this particular pathway. The plants use a different mechanism than this, but the bacterial enzyme was able to actually carry out both of these steps. And then the last enzyme, Lycopene Cyclase, is actually interesting because it turns out that the rice itself does actually have the Lycopene Cyclase sufficient to promote eventually synthesis of beta-carotene. So the idea was to engineer the plant so that it contained this entire pathway so that beta-carotene would be produced. And these molecules are very terrifying looking in terms of what they have, but they make sense. Okay, so the phytophytoene synthase was from daffodil. Phytophytoene desaturase, CRT, was from a bacterium. And as I said, this carries out two steps in the pathway. Lycopene cyclase is also actually from rice. It sounds simple, but it was definitely not simple at all. Okay, so here's one version of golden rice 1. And you notice it's actually missing, well, you'll notice shortly, it's actually missing the lycopene synthase, because it turns out it wasn't actually necessary. Okay, so what's in here is two antibiotic resistance genes, the MPT2 and the APH4 for the plant, for sure. And it's got a, phytophytoene synthase is being driven by an endosperm-specific promoter. Remember that rice is essentially endosperm, and you want a gene which is very active in that tissue to drive expression of this phytophytoene synthase. Okay, now the bacterial enzyme wouldn't have the right signal sequences to get it into the chloroplast, so it actually has a transit peptide on it in order to provide the function of getting that protein into the chloroplast where the rest of the pathway occurs. And that is also driven by a strong plant promoter. Okay, so it's got the essentials, not the lycopene synthase, but it has the two critical enzymes driven by promoters which should work very strongly in the plant. It also, of course, has a left and right border derived from the TI plasmid, the original tDNA of the TI plasmid. So this is what I just said, endosperm-specific promoter. There are terminators that were originally part of the tDNA, etc. So the antibiotic resistances are hygromycin resistance, which phosphorylates hygromycin, and another, which is another aminoglycoside phosphotransferase. So it's got the pathway enzymes, pathway genes in there ready to go. So disappointingly, some transform lines made detectable amounts of beta-carotene, but not enough to be completely useful nutritionally. Some is better than nothing, I think, but it was clear it wasn't going to be enough to provide enough vitamin A for people to survive without other sources. So notice the tDNA did not have the last enzyme, but yet the kernels, or the rice, was actually yellow. And if you look on the HPLC trace, there is, in fact, beta-carotene being made in those plants. So that enzyme was actually already there in the rice endosperm, fortunately. Okay, so the other version actually included two different TI plasma, well, two different tDNAs. Again, you've got the left and right border. You've got your endosperm-specific promoter. This one is actually driving the lycopene synthase, which it turned out we didn't actually need. And you still have to have your antibiotic resistance. So these two were used together to produce the phytophytoene synthase and finally phytophytoene desaturase along with the lycopene synthase. But it turned out we didn't need those because the other molecule without it was able to provide that function on its own. So I think this is what I said. They were transformed together and they all did work. They worked somewhat, but not enough to really treat the vitamin deficiency. So this was a successful proof of concept but not successful enough to be sending it out to the farmers or even using an adequate field test. Okay, so the version 2, which is around now, works better. And the large main reason is that instead of the daffodil, we're using corn to provide the phytophytoene synthase gene. So the corn enzyme actually worked much better than the original daffodil one. And there have been a few other minor changes in terms of the selectable marker, etc. We're still using the bacterial enzyme for the middle parts of the pathway. And again, we're still using the transit peptide from the that's actually from petunia, to drive the CRT enzyme into the chloroplast. And the endosperm-specific promoter is actually being used on both of those genes. Okay, so this version is successful. It makes 30 times more beta-carotene than did the original. Okay, so here's a picture. GR2 works well. The wild type, of course, is white, and then the original one was pale yellow, but the one with the corn version of phytophytoene synthase is nice and bright with plenty of vitamin A for any useful purpose. So this should be a triumph, and people should be willing to use it. In fact, a lot of the strains that have been, one of the objections really is that a lot of strains are adapted to the local environment, and it turns out that this has been easy to transfer to those strains, and so the local strains can actually be made to produce the vitamin, or to produce the beta-carotene. The inventors have donated the patent, so nobody's going to make mega bucks on this, and the seed is going to be inexpensive or free. And of course, we're continuing to encourage people to eat fruits and vegetables as more natural sources of vitamin A, but that's not always possible. So starting off in the Philippines this year, we'll see what happens, whether this is widely adopted or not. On the other hand, things are still controversial. There was a lot of backlash and pushing back about this in the Philippines. So we'll see what happens as a result, and we hope that it comes out that this long and successful project is able to help people. We can hope for that.